And so when I think about an integrative research environment, I'm going to introduce you to our next speaker, who is Dr. Dave Segal, who has been part of the Angelman community for far longer than I have as well. He came into this at the beginning with Ed as part of the Fast Fire team. And the Fast Fire team, as most of you know the history, really started in about 2012, so well before my daughter's diagnosis. And they were a team that Ed and Dave and Scott Dindo and a bunch of others came together, and they said, we have different levels of expertise in different areas, and if we work together, we're going to do things a lot faster than if we work independently. And so Ed was really the father of that, and Dave, you came in right after, and together they really made some incredible progress. So he's going to talk to us about the tail of that progress. This started a long time ago. He's still at it, so let's hear where he is now. Dave.
Good afternoon. I think it's afternoon. It's a great honor to be able to talk to this audience again about our progress in trying to develop these therapeutics. It's true that Ed and I go back a long way in trying to develop these kinds of therapeutics, and it's just been amazing to see the growth of things and how things have turned out up to now. I don't think we had ever imagined at the start of this that there would be so much, not just in therapeutics, but so much infrastructure and growth in every dimension of how Angelman therapeutics could be put together, ranging all the way from the things that I'll talk about today, making therapies, to accelerating the therapies, to all the way going up to the Capitol and lobbying them to make a better environment for us to make these therapies for Angelman syndrome. So it's just a tremendous effort that FAST has really put together, and it's just amazing to have been part of this. But I'm still part of this, so we're still working on things. So this is my disclosure. I do some consulting. And so the kinds of tools that we're talking about in this session are what I would call molecular therapies. And to explain, kind of the background of molecular therapies, small molecule drugs, the kinds of drugs that, people might take in a pill, typically work on the level of proteins. And since this is a genetic disease, small molecule drugs have not been as effective in trying to treat these kinds of genetic diseases. However, with advances in the last several years, now we have the ability to treat them on a kind of RNA level. So that would be things like antisense oligonucleotides are acting at the level of RNA. And then things like gene therapy, CRISPR editing, epigenetic editing, work on the level of the actual DNA, where the defects are. So molecular therapy is really getting at the molecules, the DNA itself, and trying to fix that or adjust that in some way that we could use that as a therapy. So I'm going to tell you about a couple of strategies that our lab is taking to try to develop therapies for that. And one molecular therapy that I think everyone is familiar with is this antisense oligonucleotide. So the antisense oligonucleotide binds to that long blue transcript. So I think we've heard Allison and Ed talk, and Dr. Baudet talk about this very long antisense transcript that somehow keeps UB3A kind of in an off state. And that antisense oligonucleotide binds that RNA kind of in that position and just chops off the end of it, and that allows the UB3A gene to just come on by itself. So this has been fantastic, and the results of using this ASO has really altered the history of this disease. Let's put it that way. We are thinking of a therapy that's kind of based on this, and Ulla Baitner in my lab has been heading this approach, where we want to introduce a protein called Cas13 that will also cut the DNA or cut the RNA at around that same place. So you've probably heard of CRISPR-Cas. CRISPR-Cas works on DNA. This is a kind of cousin of the Cas system, Cas13. It's another enzyme. It only cuts the RNA. So we can target this Cas13 to a very specific site on that long blue transcript, and when it cuts it there, it will actually do the same kind of thing that the antisense oligonucleotide has done. What we think is the advantage of this is that this should be, a quote, one and done, because this way we put it in as a gene therapy, and instead of having to take the antisense oligonucleotide maybe every 4 months, hopefully, if things work out well, this should be something that would last a lifetime. So that's the tool that we're putting in, is that Cas13, and that's going to cut that RNA. But there's 2 parts to always these molecular therapies, and so now I've just talked about the cargo, but we would also talk about the delivery system. And you've heard a lot about AAV already. I would just say that this adeno-associated virus is the kind of go-to virus for trying to put molecular therapies into cells. Delivery is one of the hardest parts of trying to make these therapies, but AAV does pretty well. In the early days, we had to inject directly into the brain. And in these brain slices that you see in this image, the little black parts are the parts that were affected by the AAV, so not so much. But nowadays, there's been new breakthroughs in making versions of the AAV that are actually able to get the brain much more efficiently. So you can see in this panel in a mouse that the more standard is kind of dark, and then there's a bright green one, and that's the new version of the AAV vector. And this was came out a little while ago, but now they even have ones that work in humans. So this was something that we always thought would be coming, and now these things are here. So that's what I mean. This is a very evolving landscape, and we try to take advantage of these latest advances. And so the idea is that we could use this kind of vector and get it into the brain, in human and in mice. So in this project, we did inject this Cas13 using one of these more advanced AAV vectors to put it into the tail vein of a mouse. And when we did that, we actually did see an increase in UBE3A. That's that little kind of graph thing in the corner. And so, so that was very interesting to us. And then we were in the process of trying to do the other kinds of behavioral studies that most people would do, that the kinds of things that you've seen here before. And then a very interesting thing happened. We actually kind of got scooped on this. So a group in China led by Hugh Yang published a paper using pretty much the same enzyme to do pretty much the same thing. They were able to show that they could get very good results with this, that they could turn on UBE3A in the brains of these mice. And I'm not going to go through all their data, but just to say that they did many of the same kinds of tests that we were planning to do, and were able to show that they had a benefit to it. And so at first I was thinking, oh man, that's exactly what we were trying to do. And then I thought, well, actually, they just did us a favor, because now this already shows that this approach can work, and it also shows that people around the world, more or less, are thinking about using the same kind of approach. So I think that this kind of shows that this is a very sought-after approach, and that the data supports that this can work in mice, right? So that's good. What we're trying to do now in our lab is to make the human-targeted Cas13, so not something that works in the mouse, but something that would work in the human. So Adele Guglielmi is a project scientist in my lab, and she just gave a talk yesterday about the work that she's doing there. But basically, we're trying to work on coming up with a version of this that is going to be the most effective, guided to the correct place on that long blue transcript where the scissors are. And so what I'm showing you here is a whole bunch of so-called guide RNAs that are guiding it to different parts of that long blue transcript. And the ones with the red arrows are ones where it really reduced that down to almost zero. So that's exactly what we would like to see. And as you see, we have many candidates to test, and we're going to be testing them very soon in neurons, in human neurons. And from there, we want to look at safety tests. And I really think that this is something that we can try to push to an IND level in the very near future. So, no, not yet. So now I've told you about that part with Cas13 that we're taking for one plan. Another way that we could try to turn on Dad's gene is to come in with an artificial transcription factor that would shut off that long blue transcript earlier on, or at least knock it down, right? And so this we're using a different type of tool. It's called a zinc finger artificial transcription factor. But all you need to know is that when this thing lands in a particular place on the DNA, it stops, it reduces the level of that transcript. And again, that should allow UB3A to come on. So these, similar kind of approach. So for people that have that may remember some years back, this is an idea that we kind of published on before. But back then, we were just injecting the protein. Now we're putting this in with one of these AAVs. So this actually worked a little bit better for us. So that's the difference in what I'm talking about today. And so, we want to get that whole brain delivery. And I think Allison showed this slide before. We were able to inject this version of our therapy into a mouse as well. And I want to emphasize that this is like an older mouse. This is 5 to 6 weeks old, so a kind of more mature mouse. We were able to knock down that long blue transcript, not all the way down, which we think is important. So there'll still be, the parts that need to be spliced out for brain behavior. We think that will still be happening, but we knock it down enough so that UB3A can come on. And we could see UB3A coming on in these mice. So again, just if you look at panel D there, on the left side is the wild-type mouse, in the middle is the untreated Angelman mouse, and on the right side is the treated Angelman mouse. Looks a lot like wild-type. So we're turning UB3A back on. Now, does that make a difference? So, you've seen a lot of these assays now. So we looked at this open field assay. All I really want you to pay attention to this. The black line is the wild-type mouse, the blue line is the Angelman mouse, and our red line, the treated line, is a lot more closer to that black line, so a lot more like wild-type than it is like Angelman. So that means, that we're doing very good at rescuing some of these motor phenotypes. We also have the rotor rod. That red line's kind of in the middle, so we didn't get a full rescue, kind of a partial rescue on that, but still looking very good. And so I think, with these things, it's looking like, it's working pretty well in the mouse. We also did some other experiments that I'm not going to show, trying to look at cytokine and inflammatory markers. Everything looked very good. So we think that this is an approach that could also be translated into human. And so again, what we're working on now is trying to make the version that would work on the human genome sequence and not the mouse genome. And so we already have a factor that looks like it's kind of repressing just like we had in the mouse, but now this one is targeted to human, and we're just going to try to work that up and show that can do the same kind of thing in human neurons. So in the end, in summary, I've tried to show you 2 different ways that my lab is working on to try to turn on Dad's gene. And you might legitimately ask the question, well, why don't you just pick one and do that one? Why don't you just pick the best one? And I just want to leave off on that point that many times I think in therapy, we try to have a very clear idea of what we think the ideal therapy will be. But I think that it's actually better that we try many different approaches. Like they say, take more shots on goal. Because the more shots on goal that we could take, the more likelihood that we will get something that is a real winner. So my feeling is I don't want to pick the best horse. I want to work with all the horses, and we'll just see which horse wins. And whichever horse wins, that's the therapy that I think would be best for your kids. And I will stop there and thank the people in my lab, and thank you.
Any questions? I see one coming over. Right here. Go ahead.
Hi, David. Sorry, it's me again, Emma. Do we know why Dad's UBE3A is silenced in the brain? And does the ATS have another function other than just to silence the paternal UBE3A?
So yes, we know that that gene is silenced in the brain. And there does seem to be some parts of that long blue transcript that get spliced out and seem to have a role in healthy brain functioning. So if we chop that off close to where the UB3A gene is, like the antisense oligonucleotide does, those parts of the transcript seem to be in place. On our other therapy, we're trying to knock down the level of that transcript so it's not completely off, and so those parts will still be made, but it's knocked down enough so that UBE3A comes on. So we try to take into account that there may be some functions that are necessary from that transcript, and we're preserving those in both of our approaches.
I see one coming over.
Thank you. If I understood correctly, with the CRISPR Cas13, you only, unsilencing just a small piece of that ATS and just the UBE3A, but with the zinc finger, you would do that earlier on. Mm-hmm. So do you see an effect of now unsilencing SNRF, NSS, and RPN, the other gene? Do you see an effect then on the mice?
So the SNRF gene, we think, is one of the primary origins of that long blue transcript. So I think that gene was already on. And so in the second approach, we're kind of turning that down, but not off. And so at least in the mice, that seems to work very well. And like I say, it should preserve all the functions of the little pieces in there that are needed.
Okay, thank you, Dr. Segal.
Thank you.
Very scientific and very exciting, and hopefully the take home there is those were one lab doing two paternal activation strategies that are different. One is being very targeted in terms of targeting a sequence just upstream from UB3A, and the other is much less targeted, targeting an area by SNRPN, to your point, Samantha, which I think is really important. The impact that could have on the SNO RNAs, which are related to Prader-Willi syndrome, in mice seems to be okay. We don't yet know the impact of that on humans, but it doesn't look like it's knocking it down completely to result in a Prader-Willi phenotype or to result in a Prader-Willi genotype. But it's something to think about. And this is the whole point of exploring, as Dave said, everything, looking at all shots on goal and trying to understand what has the best result. Sometimes you don't know until you try, and this is the opportunity that we have, is to try all of these options in mice and really compare them head to head, like I showed you in that heat map, to say, what are the risks, what are the potential benefits, and then some may ultimately turn out to be no-go decisions because of toxicity. So then you have to do all of those studies for toxicity to say, what does this look like in a different species? What does this look like in human cells? Can we get from mouse candidate to human candidate and accomplish the same goal? And so these are early days for a lot of these studies, but we're so far ahead of where we were only 3 years ago on all of these strategies, and that's because if we only had one option, we would only have one option. And we don't. We have a lot of options, and that's because we're lucky to have an elegant set of genetics that we can work with and try to find the best option. And there's probably not going to be one best option, right? It's probably going to be a little bit of this and a little bit of that and different things for different people. But I do think it's important to realize the brain went from no color to a lot of color. In all of these strategies, whether it's gene replacement pillar 1, whether it's paternal activation pillar 2.